Thanks, Miguel. All right. Good afternoon, everyone. I will preamble my talk by a excuse. I'm not a historical biologist. In fact, I am a paleontologist, like a deep time paleontologist working with Eocene and Miocene insects. But I also work with extant insects, and then I find out that something is missing. So hence I'm here. Another preamble, I just started my Ramon Cajal project here in Doñana. So a lot of things I'm gonna be talking about are plans, not concrete results. But with that, let's start. So I want to start with, first I have to thank to Graciela and Adam for their excellent talks on paleolimnology, essentially. I don't need to give you a primer of paleolimnology anymore, so half of the slides are going into the bin. But I want to start with a short comic, which I produced together with my colleague Valentina Inshina, who is here in the audience today, on the history of interactions between anthropogenic pollution and fresh waters. So humans first started really interacting with consequences of polluting fresh waters when probably Neolithic agricultural revolution happened, because, you know, humans become sedentary and their waste were staying where they are. And that was having consequences: increased exposure to the parasites, increased exposure of the domesticated, semi-domesticated mammals and birds to the results of pollution and so on. But what's interesting for me is that this pollution left a trace in the geological record. But what is even more interesting for me, I'm aquatic insect ecologist, paleobiologist, that we really started affecting how the aquatic biota lives at this point. You know, beyond just fisheries and aquaculture, our pollution become a type of interaction of humans and aquatic biota. I mean, with development of complex societies, the pollution ramped up. It is well-known fact that use of the lead piping in the Rome and other big Roman cities has left geochemical traces in sediments, and you can trace how certain cities were Romanized by bringing in the viaducts and lead piping, by studying, for example, a Tiberus River bottom sediments, and so on. And not until the Industrial Revolution in Europe, when these two gentlemen, Max Schwann and Theodor Schleiden, from the Imperial German Institute of Freshwater Science, start looking into consequences of pollution, because, you know, Prussia ran out of drinkable water in 1970s, and so, like, holy crap, we have to do something about. Not until then we really started quantify the impact of the pollution. But now we know that impact is immense, it's global, it's essentially a planetary geobiological factor, and it unfortunately impacts aquatic insects so dear to my heart. And with this short preamble, the main goal of this talk is to look for possibilities to use a methods of paleobiology in application to the conservation and monitoring of modern and recent historical aquatic biotas. So I have here the whole lot of slides about what paleolimnology is and how paleolimnological studies are done, but again, thanks to Adam and Graciela, I don't have to go through all of that. However, I'm working mostly on chironomids, or non-biting midges. Those are one of the most abundant and species-rich aquatic insects in the world. There are over 11,000 species living everywhere from Antarctic Peninsula to 6,000 meters above sea level in Himalayas. Adult flies have bushy antennae. They spend most of their life as larvae in aquatic sediments or within the plant tissues or even in terrestrial ecosystems. And they spend a short pupal stage also normally in the water. Larva has 4 larval instars, so they molt 3 times. each molted skin normally stays in the water, settle down to the bottom of the water body. If it's lake and don't have a high energy sediment transport, the head capsules has a great chance to stay where they are and become a paleo-environmental proxy alongside the pollen or a cladoceran mandibles and so on and so on. So, you know, you extract cores just as for pollen, or sometimes you extract them together, and then similarly as with pollen, you deflocculate your core, extract your specimens, and work with them. That's how some of the more animal paleolimnological proxies are looking. We were talking a lot today about pollen, but so this is a Cladocera, this is Daphnia pulex. This is a chironomid head capsule. That's another two chironomids, and this is a Cauberus, so the phantom midge. Those are more abundant and frequent groups of the freshwater organisms used as a paleolimnological proxies in the lakes. You know, this is how the chironomids are looking like. They occurring in the lakes in huge numbers, and they also occupying a variety of different niches, and they have a, well, sometimes very specialized larvae which can only tolerate narrow environmental conditions. Most importantly for paleolimnology, temperature, because chironomids are frequently serve as a basis of the paleo-temperature reconstruction for the Holocene and the latest part of the Pleistocene. We can do it because there is immense amount of chironomids in some lakes. For example, the lake Mjøvatn in Iceland, which is translated from Icelandic as midge water, can have up to 200,000 specimens of chironomid larvae, which normally look like that, per 1 square meter of the bottom sediment. So that's quite a lot. And they're also important ecosystem engineers. To give you a perspective, a Lake Müggelsee in Berlin, which has a surface water area about 15 square kilometers, is getting pumped through the burrows of little chironomid larvae once every day. So that's about a half a cubic kilometer of water going through the chironomid burrows every day. So they are important, they are abundant, they are everywhere, and that makes them excellent environmental and paleo-environmental indicators. That's the head capsules, essentially the proxy that the 4 instars of chironomid larvae are leaving behind. Okay, not the first instar, because they have a very soft, non-sclerotized head capsule, but 3 next ones. Okay, so some more of the head capsules. Those are actually subfossils, and these beauty shots are from the extant ones I collected in the Alamillo Park here in Seville. So they're in actually much better shape than the subfossil ones would be. And when you go through your core and extract chironomid head capsules from sliced intervals, identify them normally not to species but to the larval morphotypes, assign trait scores to them, you can build reconstructions very similar to what Graciela demonstrated for all other paleolimnological multiproxies. It's normally done for the temperatures, but you also can reconstruct environmental parameters such as eutrophication, nitrate loading, water levels with chironomid subfossil communities, and it works well with Holocene and Pleistocene, with chironomids, just as with the plants, because we have essentially an array of extant taxa whose autoecology we can observe directly, and so we don't have to guess what is their environmental tolerance and so on and so forth. That's how subfossil chironomids are normally used, but that's not what I'm going to be talking about today. You know, so normally we're using chironomids to reconstruct the paleo-environment and paleoclimate around the time of last glaciation. Like some very cool reconstructions were done at the Wrangel Isle, where the last mammoths were coinciding with pyramids being built in ancient Egypt 5,000 years ago. Here is an excellent illustration by Ukrainian paleoartist Roman Uchitel. But what I'm going to be talking about today is about how we can use chironomids and other subfossil insects to reconstruct recent human impacts on the environment predating available monitoring data. So, as I said, besides being a paleontologist, I also work with extant aquatic insects. And when I was a postdoc in the Senckenberg Museum in Germany, I got to work with one of the best and longest aquatic macroinvertebrates stream data set from a Schlitz River station of Max Planck Limnological Institute. It was a daily effort on collection of aquatic insects, which ran for 42 years. So it's extremely high resolution data set. And Institute was employing several insect taxonomists, at least one person who was proficient in identifying mayflies, one professional for stoneflies, one for caddisflies. Those things doesn't happen anymore. Unfortunately, it was closed in 2010, but we got a data set to work with. And that's how the daily catch of the aquatic insect looked. We also got some very good high-resolution environmental data. We got a significant increase in the mean annual temperature over the 42 years of observation, almost by 1.8 degrees of mean annual temperatures. That's outpacing the average rate at which our world is heating up. And also here is the hydrological regime change. You see that light green is extremely dry years. So stream has transitioned to extremely dry years as a dominant pattern of hydrology over the period of observation. And on the backdrop of that, we got this graph. This is a change in the mean annual abundance of aquatic insects in the stream in thousands of specimens per year. And we transitioned from approximately 30,000 specimens per year to less than 10. So we got a 80% drop in 42 years. And this paper made some media rounds. And I think a lot of people overread it. So it's a little bit like Holman's et al. Krefeld study, where biomass of flying insects dropped by 75% in 30 years. And it led to, to a pervasive rhetoric of Insektensterbe and Insektarmageddon, and insects are clearly declining. It's just, all our debates are limited by the range of data that we are really having. I mean, there are all kinds of anecdotes like windshield effect, and we clearly know that genetic diversity in all the insects for which we examined in ancient DNA seem to drop. The question is, however, I have this excellent data set. I might extrapolate it into the future and say that by 2020, aquatic insects would be gone from Breitenbach, but that didn't happen. I've been to Breitenbach, still has some insects, although I didn't sample. The question is, how can we go beyond the monitoring? How can we stop extrapolating and speculating and actually get some data? So I drew this graph in paint, so in solid red, we have our monitoring data. That's what we have. That's what empirical, that's what we can argue about. But what have happened before, we don't know, and that's actually impacting our ability to forecast. What will happen next? Will the downward trend continue? Is it just a part of a cycle of boom and bust in abundances? Or will it maybe, as some right-wing politicians in the area were telling my colleagues, it will just, rebound and go back because it's great. Can you see that everything is green here? I forgot to tell that all this data were collected in the nature preserve, which never got much of any direct human interference. We don't know. What can help us to forecast better is more historical data, but obviously we don't have more monitoring. We cannot will it into existence. What we can do in some cases is to go for the paleoenvironmental proxies. Say, sample chironomid core for the lake for which you have 30 years of monitoring, but that's it. But instead of looking into the Holocene climate, try to look to the rank abundance over the last several meters of sediments, which probably will represent several hundreds to several thousand years, depending on the rate of sedimentation. And so, yeah, this is just to show some pretty deep time fossil pictures. As I said, I work in a deep time, and I was working a lot with extinction dynamic of aquatic insects in Cenozoic and Mesozoic. And I was feeling that extinction dynamic was very different from what we're seeing now. And that was my rationale to actually start this Ramon and Cajal group here at EBD. Come and see if I can plug this missing data record with using paleobiological approach to essentially modern ecosystems. From fossils, we are getting a whole range of information. For example, we found this piece of amber where we had a 56 fly larvae on the piece of feces of mammal in Baltic amber. That was the first undisputed mammal record from Baltic amber, albeit feces. And it told us a lot about the Baltic amber ecosystems that we didn't know from other sources. Because, for example, we find out that there was enough open canopy areas where the mid-sized herbivores could munch on the grass, leave feces, and attract the saprophagous flies. And if we can learn so much from a piece of amber, I would argue that we probably can gain enough information from paleobiological evidence for the several last 100 years. For example, here is a Corona larvae, which is 38 million years old. It's also from Baltic amber, and it's showing exactly the same commensal behavior with a Leptocerida caddisflies by sitting on their case as the modern ones. So, we can get a lot of resolution and a lot of data power from a very old deep time fossils. And so I was, okay, let's see what we can do with something younger, with something even younger than Holocene. And so, I'm presenting what I'm just hoping to be doing. So I came here 3 months ago, being attracted to EBD because it's obviously an excellent institution to study wetland ecology, having Danyana, which I hope most of you will see on Saturday, but also because southern Andalusia is a region with such an ancient human footprint. Also literally, we have over 200,000 years old human footprints on the beach in Matalascañas. And Cadiz, or Gader, is one of the oldest continuously occupied cities in the world. And I thought, well, what can I do with a paleolimnological evidence derived from insects in such a storied region? So the idea here is to take the sediment cores from the most stable wetlands and lakes in Andalusia region, extract chironomids, look at the rank abundance, because obviously the concentration of head capsules in sediment cores don't give you a true abundance. But it gives you rank abundance, and that's how, after all, we reconstruct the changes in the community reflecting changes in the temperature. And to see if these changes in rank abundance corresponded to any change in agricultural and water management practices, and if maybe some species didn't survive last several hundred years in the region, probably due to the local climate change, and to see how these changes in the communities and rank abundances are coinciding with local and global climatological records. I'm now started up with building a database of the extant chironomid taxa in Andalusia, which is quite distracting because I keep bumping into the undescribed species, which holds me for a day, and then I remember that it's not the point and just shuffle them somewhere for my students to deal with later. What is also cool about using chironomids as a proxy for status of aquatic communities, for abundance of aquatic insects in the not such a deep historic past, is that they can go beyond just a taxonomic proxy for any kind of environmental conditions. They are also quantitative proxy. We wrote a paper with my colleagues where we showed that there is a very strong linear interaction between the body size of chironomids and the ambient mean annual water temperature. Essentially, for every degree, for increase by 1 degree Celsius of mean annual temperature, chironomids are becoming 32 micrometers smaller on average in the community. And you can see that orange line represents this dependency for the northern hemisphere and black line for the southern. We think that this, and flies are obviously getting smaller when it's getting warmer. This is almost like a Bergman rule type interaction. But of course, chironomids are poikilotherms. We think that the limiting factor here is a lower solubility of oxygen in the warmer water. And what I'm trying to say here is that by studying near-time historic records of chironomids, looking and comparing the size of the head capsules with the calibrations which I'm currently trying to produce, both in the lab and in the in-field experiments, you can trace the changes in temperature not by just looking at the community composition, but also by looking at changes in size. So this is just a map of the chironomids we used for our initial size temperature study, and you can see distribution. The color is size in micrometers. Yeah, this is my lab setup where I'm currently trying to run a culture to calibrate the rate of size change for the different temperature and hypoxia level. But this is basically was just to say that I have some ideas and I'm interested in cooperation. This project has just started, but I have to say thanks to Spanish Agency for Innovations who's funding Myra Monica Howe, but especially to all my colleagues, to my PhD student Caro Cimeno, to Oliver Haley from University of Basel, who helped me to bounce up and refine lots of these ideas, and to many other people. And I wanted to say thank you to Carlos Villa, Jennifer Leonard, and all other my new friends and colleagues in EBD who made me so welcome here. And I want to say special thanks to Roman and Alexandra Uchitel, amazing Ukrainian paleoartists who allowed me to use their art, and just to use this platform to say there is amazing paleobiologists and paleoartists in Ukraine. Look them up. Thank you very much.
Thank you so much, Victor. Before going to the questions, we have to say that we are going ahead for the accumulated delay. So we are, after the question, we are going for lunch, and we will be here, if possible, again at 2:15. Just try to be on time for the archivo. If you don't mind, Adam, we pass it after the lunch. I don't know. Okay. So if there are questions for Victor.
Thank you so much. This is all new to me, so it's fascinating. You explained it so well. My question is about the idea of insect Armageddon. And I think as scientists, we are always trying to present the uncertainty and the caveat in what we our results. But then, is it better for the public to believe that there is an insect Armageddon, even if it's not entirely true, or to say, oh well, maybe no, we don't know how humans are affecting?
I don't know how to answer this, Molly. It's like your green list, will it make some people believe that some species are doing better and don't need further conservation effort? I don't know. So I'm a vain person. I was looking after altmetric of this insect decline paper for Breitenbach, and I've seen that Bayer, so the large chemicals German company, used this paper to justify that insecticides became less deadly for aquatic insects over years. I'm not sure how did they arrive to this conclusion. It's like as a scientist, when we're putting something out into the world, we don't control the interpretation, no matter how much psychom and explaining we do, I believe. So maybe it's better for the situation is dire. I just, my idea here is not to say, oh, maybe everything is great and we just need more paleobiological data to prove it. No, so like Professor Chase here published an amazing meta-analysis showing the complexity of the processes going into the decline in different systems, in insect populations. And I found that this complexity is sometimes difficult for public to get, and it leads to more levity than it probably deserves. But I'm also don't feel comfortable telling people, you will die because there will be no pollinators in 2 years.
Thanks, Viktor. This was really fascinating and inspiring. The last time I heard about midges was my time in Wisconsin Madison, where Tony Ives was presenting his work at Lake Mývatn in Iceland. And I remember he was mentioning these interactions between sort of midges, production, and human pressure, but he was talking about it the other way around. Midges are the ones that are fertilizing the soil and making the grasses grow, and that increases grazing pressure because there's more farmers bringing their... And what I think I heard you say is the opposite, that you want to measure use the midges as an indicator of human impact, or do you want to measure the human impact on the midges? This was unclear to me.
So I want to measure midges as an indicator of human impact, but Lake Mývatn is a unique system. It was used, so Tony Ives together with Zaten and Grattan group from Minnesota wrote this amazing paper on the non-linear states in the ecosystems based on chironomids in Mývatn. Mývatn is amazing. So there is this four-patriate system between the migrating birds, midges, grazing cattle, and the riparian arthropods. Ducks are coming, bringing nitrogen, fertilizing the lake, more chironomids, fertilizing the riparian zone, more insects, more migrating birds are attracted, and then there is also humans and the grazing cattle. Normally, chironomids, while taking part in the energy matter transfer, so they are so-called uphill elevators because they transfer nitrogen and phosphorus in particular back to the terrestrial system from the water, because as most of you know, phosphorus normally going one way downstairs. What's in the water stays in the water, and then getting, especially if it's in the ocean, is just getting lost with a crust subduction. And chironomids returning it, but normally they don't fertilize riparian zone nearly to the extent of Lake Mývatn, because to give you an idea, Lake Mývatn has 200,000 specimens per square meter. The average Central European lowland lake will have 1,000 there. So these types of impact are rare. The other one which comes to mind immediately are not chironomids, but phantom midges, cowbirds in the Lake Victoria, which are getting baked into the kunga cakes by local population and are a food staple, in fact. And they also fertilize the riparian meadows around Lake Victoria. But normally it's going like that. Humans come in, humans increase in nitrogen pollution, 90% of species richness of chironomids are gone, Chironomus riparius appears, entire community getting replaced, and hey, you got a clear break and shift in your sequence. So, yeah, but in fact, the idea of midges influencing human societies is fascinating. Just a very, I'm sorry, we're going to lunch now, I promise, but I have to tell about it. So when Brits built an Aswan dam in Egypt, a lot of the local people start suffering a very atypical for the region respiratory disease. And it becomes so problematic that even British colonial administration had to react. And they sent young Paul Freeman, erstwhile curator of Diptera in London Natural History Museum. He spent there some time and find out that damming of the Nile caused Cladotanitarsus pseudomangus, my favorite pet midge in Africa. It's everywhere, I always find it. It's striped like a little tiger. So the damming of the Nile makes them bred in the huge numbers. And midges, to survive in low oxygen water, has a larval hemoglobin. This hemoglobin is very different from ours. It's a super oxygen-affine hemoglobin. And it's very strong allergic ligand for many people. And so all these Sudanese there got a bronchial asthma as a reaction to the overproduction of the midges, bringing a hemoglobin to them. So there are cases when human society is getting impacted by midges, but normally we're just screwing them over.
Thanks so much, Victor. I'm looking forward to seeing the results of your work in Doñana. We talked about this briefly before, but I'm very interested in the potential of chironomids for indicating the presence of, and maybe the abundance of, pasturing animals on a landscape. And I don't quite understand the mechanics of that, and I'm just curious if you could maybe talk a little bit more.
Yeah, absolutely. So there are two elements to this mechanics: geochemical and your OD looking into the stereo microscope. So there's a basic level of this mechanics. You're looking at what taxa, morphotypes, because we're normally not capable of identifying fossil chironomids precisely to the species. We're identifying them to species groups. And you know that, I don't know, so your Tanitarsus gregarius, which was a dominant species before year 1600, declined and then disappeared, and Procladius, Hareus, and Chironomus, I don't know, Pseudotumi, become increasingly abundant. And we know that the later two species are alpha mesosaprobic to polysaprobic, and the former is beta mesosaprobic to oligosaprobic. So it means that the organic loading on the water body has increased. That's one way to do it. There is thankfully more technological way to do it, developed by Oliver Harris' group in Basel. So, there are carbon isotope fractioning based on where the carbon is coming from. There is a big difference between autochthonous and allochthonous aquatic and terrestrial carbon, but you also can tell if carbon was coming mostly from the deciduous flora leaves dropping into the water once a year, or if it starts suddenly coming from manure. You just need to calibrate for that. You need to look at the different type of isotopes. Normally it's done in the lab, and then you basically just do an isotope analysis on the head capsules. So that's the mechanic behind it. Yeah, and if anybody has, any cores or any lakes near the places you want to look in the interesting historical events, let me know. I will be happy to look at the chironomids.
If we don't have more questions, we'll go to lunch. We will be there saying, come on, let's go back. But anyway, if we don't do it, try to be here at 2:15.